Drug discovery pipeline is a multi-step process which starts from genome identification and ends at distribution and post-distribution evaluation. Now, four major steps of drug discovery are target identification, lead selection, then clinical trial selection, and then clinical trials. Now, what are different strategies for discovery of drugs? So to answer this thing, we should have two major constituent parts. Number one, that our drugs should be made through computerized technologies. So when we say that computation is required for making something, we say so because it would be very efficient and it would be done on a very high scale. Then we should have a good information or understanding of structural information of different molecules which are being used for generation of drugs, and then also for understanding of molecules inside the organism body. So drug discovery can be done at two levels, and those levels are either genome level or proteome level. So genome level is directly related to genes information, and proteome levels are related to proteins information. So we will start from genome technologies. So for genome technologies, there are different type of information which can be done or which can be used for discovery of drugs. So we start from basic DNA sequencing, which we said novel DNA sequencing. Then we have DNA microarrays and chip technologies. So novel DNA sequencing is dealing with single DNA sequences. When we say DNA microarrays, then we are moving from more than one sequences. Then we have metabolic profiling. So metabolic profiling, where we have converting DNA sequences into some other chemicals or other information. So what does Human Genome Project gave us? It gave us 30,000 genes, and then the study of these genes help us to identify different diseases, and for those diseases we invent different drugs. So at a first look, this 30,000 genes looks very small amount or very small number for drug discovery. So what happened later on, that we had the effect of alternate splicing. So this alternate splicing, it is basically a concept which says that same genes can be transcribed into different proteins. So 30,000 genes, they are transcribed into different proteins based on this alternate splicing mechanism. Similarly, we know that 99.9% of human genes are similar. There is only a difference of 0.1% genes. So this difference is enough to discover new drugs and try these drugs on different diseases. Here I have given an example that genome-based techniques or gene-based techniques can be applied to a cell at different biological levels. So different biological levels means that we can start from gene level, as shown here in this figure by genome sequencing, this first part. And for genome sequences, we have given the example of genomic technologies. So what are genomic technologies which are available in the market to do this biological level processing? So one technology is this shotgun sequencing. This shotgun sequencing is basically DNA sequencing technique in which DNA is broken into smaller segments which are sequenced using chain termination method. So basically the idea is that the larger DNA strand is broken into smaller strand, and then smaller strands are sequenced, and then we have a complete sequencing. Similarly, transcriptome, it is basically happens when the gene DNA is converted into RNA. So for that purpose we have DNA microarrays and chips. Then we have proteomes, which are for proteins. Metabolomes can be done by different techniques such as mass spectrograms, then liquid chromatography, then NMR, which is nuclear magnetic resonance, and gas chromatography. Similarly, finally we have chemical compounds identification, which is done by screening of target-based assays. In comparison to genome-based sequencing techniques, we have proteomics technology for drug discovery. This proteomics drug discovery is based on proteins information. Now this protein information can be classified at 4 different levels, which are chemical level, structure level, topological level, and computerized level. So what are these levels? Let us see one by one. So number one is chemical proteomics. So there are 3 different things in chemical proteomics. What are major parts? So major area or major parts are that it is combination of different domains such as biochemical, mass spectrograms, synthetic chemistry, and cell biology. Then what is done, or what is function? Function is that we use probes to target proteins and different enzymes for the purpose of drug discovery. And then what are different parts of this chemical proteomics? We have our ligands. Ligands are basically our drugs. Then we have linkers, and linkers are when we link different drugs with the insected molecules. And then the taggings. So tagging are basically pointing some labels or names to different ligands and linkers. Then we have structural proteomics. Structural proteomics is based on the understanding of underlying structure of a protein. And in most of the cases, this underlying structure is a 3D structure. So this 3D structure of proteins is basically used for finding the relations which are present in the form of amino acids. Then we have computational proteomics. Computational proteomics, it again provides us information about the 3D structure, but it is in the by the help of computers, so it is very efficient, and we can deal with millions of proteins in the same way. Then for that purpose, there are several databases available, such as we have Protein Data Bank, PDB, and then we have 3D Genomics. So 3D Genomics is basically a database which provides us information of proteins in the form of 3D structures. And finally, we have topological proteomics. So topological proteomics tells us protein structures within a given cell, and we have quantitative and basic organization of proteins. So quantitative means that how many it is dealing with the number of items in the protein, and for that purpose we have a library called TopNet which provides us information about the quantitative structure of a protein. So to conclude, we can say for drug discovery strategies we have two major constituent parts. One are based on genome information and the other one is proteome information, and major focus today is being given to proteome based techniques which are further divided into different categories.